Hello, Dr. Gittler.
Hi.
Oh,
Hi, Peng.
hi. How
Hi.
are you?
Good.
Good.
Oh, we both have gray hoodies on today.
Yeah.
All world.
So we today we just closed the shut down the campus. So this morning kind of a rush. Sorry.
Again? Yeah. Wow.
Yeah. So it's kind of My husband is online to give lecture, so I need to send the kid to school.
Wow.
Today is rainy. Kind
Mm.
of. Oh my God.
Feels
How
like
are you?
2021. Feels like
2020.
feels like 2020.
Yeah.
So what campus are you on?
I'm in the Zhejiang. You visited last
Oh,
time.
yeah.
Yeah.
Okay, so that's all closed down. What about the kids' school? Is that also online or no? Not yet.
The undergraduate student are in their dorm, so they will take the lectures online. But the kids still the primary elementary and the middle school maybe still open the campus. Yeah.
Mhm.
Yeah.
Okay.
Yeah.
Yeah, I
So,
just left. I'm in Philadelphia right now.
oh, you're in Philadelphia. You moved
Yeah.
there? Oh,
No, I was in Shanghai on Saturday, but then I came here.
oh, you just came. Okay,
So do you want to test your slides?
sure. Let me try. You can see. Can you see my slide?
Yeah.
Let me full screen.
Yeah, looks okay. Churchill, good. Stalin.
Is that smooth?
Yeah, looks good.
Okay. Oh.
Yeah, looks good.
Okay. Nice. So I will try to keep my talk within 20.
That would be great.
Yeah. So need to stop sharing. Yes. Okay.
How's Hailan Hu doing?
Oh, Dr. Hu, now she moved her lab to the new building. Did you know
Okay.
that?
I
They
didn't
have
know.
the Brain Institute now moved to another campus, actually.
Mhm.
Most of the faculties in the neuroscience actually moved there to the lab, but it's still in transition, I guess.
Does she still have her pet bird?
I don't know.
Okay.
Yeah.
Yeah, last time I visited, we went hiking and she had a pet bird.
Mm. I heard something about it, but you can ask her.
I will.
Yeah.
The
About
bird, the bird like just sits on her shoulder as they go for a walk.
Take time to take care of the pets.
Yeah.
So my daughter always want to have a cat. I said maybe until she can take care of the cat, otherwise we cannot afford it.
Yeah, we got a dog for the kids. They said they would take care of it, but we're the ones who take care of it.
Yeah. So everyone we can see in the participants, right? Now they're not get online yet.
Yeah, they'll come in as it gets close to the hour.
Okay, I see. So I will be the second.
Yeah.
Okay, yeah. Great.
Hi, Cheen.
Hey,
Hello.
Thanks for coming.
thanks.
So do you want to test out your slides?
Sure. Let me see. So probably have to. Okay. Screen. Let me see how it's. Okay, share screen. You guys see the slides?
Yeah, looks beautiful.
Right. Should I just leave it on, or should I?
No, I'll have like an introductory slide first, and then you'll be the first presenter.
Okay. All right.
I was actually in Boston earlier today.
Oh, nice. I was like totally. This is the first time I'm presenting at like 8 p.m. for a while. So, but it's nice.
Yeah.
So you're in Boston for a conference or something?
I was just visiting like a meeting at a company, and then
Okay.
I'm in Philadelphia now.
Okay, nice.
So what program were you in at Stanford?
Oh, I was in the bioengineering program, so I actually took one of the classes from you and actually Ron. Back then, there's like this mini courses on neurodegenerative diseases.
Yeah. Okay. Where the patients would visit?
I don't remember. It's the ones that we go to.
What year?
Oh, that's a long time ago.
Before COVID?
Before COVID, definitely. Yeah, it was organized by you, Ron Copito, and the
Reimer?
aging is in VA. Yeah. So this, we went through like multiple different degenerative diseases, and I still keep getting like emails from, and I was like, maybe I can just sign up for this one.
Nice.
Yeah.
Yeah, we would have patients visit us, like with ALS or Alzheimer's disease in the class.
Yeah. I still remember like one time you were doing the ones that do like yeast screening on like the different types of proteins, like the
yeah.
Aaron, I'm 1 block away, 1.5 block away from my one.
Okay, drive safely.
Yeah.
So it's not so cold in Boston. I was expecting it to be really cold. I bought a hat and gloves, but I didn't need it. It was 50 degrees today.
Yeah, it wasn't that cold. I think for the past few days it has not been, especially rain yesterday. So it kind of like increased the temperature a little bit. I don't know, like always I feel like when it's raining, it's not really going to be that cold. Yeah, so for the past few at least 1 week has not been that cold, but the week before that it was pretty cold, but not manageable. It's not like it's not manageable. Yeah. And then a few weeks before that it was kind of warm, so it was like almost like summer like temperature. Yeah.
Oh.
Yeah.
And then in that class, Cheen Young, remember we had like a field trip to the pathology lab, and we would look at brains and things.
Yeah. I think so. I think we look at brains, and it's just like it has been a while, and but I remember taking that classes, and I'm not sure it's also the ones that introduced me to like the. I'm not sure whether it's the years that coincide with the discovery of the hexanucleotide repeats.
C9, yeah. Did you yeah, that might be interesting to use some of your approaches to see what are the epigenomic and transcriptomic changes in patients harboring the C9orf72 mutation.
Right. Yeah. Although it's like really fresh, like if it's kind of like the technique is pretty fresh out of the the publication, and so I'm still like thinking about the way to actually present it so that it's actually can be tailored to like a broad audience, like this Zoom meeting. Yeah.
Well, this is like a job talk for you.
It's a job talk. It's a partial job. It's like because I also plan to kind of incorporate things that I did in Forensic Lab into the job talk. So it's like maybe partial. Yes.
I always prepare my talks as if every talk's a job talk.
Oh, yes.
Hi, Jin.
Welcome back.
Thanks. I'm in Philadelphia now.
Are you? Oh, are you going to ASCB?
No.
Ah.
I'm going to CSHL.
Oh, right. That's starting this weekend.
I've lost all track of time. It's starting Wednesday.
Okay. Thank you. It's lovely color.
Oh, nice. I only have the old version.
Ah,
I don't even have the new version. That's nice.
the new version so good.
Nice.
Yeah.
I think San Diego has the most number of NeuroZoom hoodies. Oh, there's another one. Wow.
Yeah.
Wow.
I have more. So that was a flat back.
Yeah, I haven't even gotten one yet because I was traveling.
Well, there's another one.
Hey, Amy.
You haven't gotten yours, Aaron?
No, I'm in Philadelphia now. I was in Boston earlier today.
Wow. It's really good.
Nice. It's comfortable.
Wow. Yeah, the new hoodie is great. I should get one.
Yeah, it's long. I'm gonna bring you
Oh,
yours to
yeah,
Shanghai.
great.
Yeah.
Welcome back.
Yeah, yours is coming to Shanghai soon.
Great. Aaron, did Peng tell you she will drop out her kids to school, so she'll get online in around 8:30, 9:30.
She's here.
I'm here.
Okay.
Yeah.
Oh, great.
I got it.
Okay, I saw it finished
I
already.
make it.
Oh, 8:30. Okay, great.
I made it.
See you.
Hi.
Yeah, I make my kids ride their bike to school by themselves.
The school bus.
Oh,
No school bus, bike. There is a school bus, but we don't let them take it.
They have bike lanes?
It's China, so not really.
So they ride on the walkways?
It works out, yeah. My kids are the only kids in China. There's like 1.7 billion people. My kids are the only kids in China who wear helmets when they're biking.
right. Should we get started?
Yes, good.
Sure.
Okay, welcome everyone to Philadelphia edition of NeuroZoom. Good to see everyone. And before we get started with talks today, here's an advertisement for next week's talk. We have Husnia, a postdoc from Brad Zucker's lab, and we have a new UCSD colleague that I bet our existing UCSD colleagues don't even know he's one of their colleagues yet. But Alejandro Chavez is an amazing high throughput screener, CRISPR expert, and he's going to talk to us about his approaches for using CRISPR and functional genomics to study neurodegenerative disease. So tune into those talks. Please let us know if you want to present your work here with us. And we'll continue to do the NeuroZoom sessions. So with that, I'm going to introduce our first speaker today. It's a great pleasure to introduce Dr. Cheen Euong. He is currently a postdoc at Harvard University with Dr. Xiaowei Zhuang, and he just had a really exciting paper, and he's continuing to develop single cell approaches to studying single cell transcriptomics and epigenomics using imaging, so spatial transcriptomics and epigenomics. He's going to share that with us. Before coming to Harvard for his postdoc, Cheen Euong did his undergraduate degree in Canada at McGill University, and then he came to Stanford for his PhD, where he worked with Marius Wernig. And here he worked, as everyone does in the Wernig lab, on neuronal reprogramming and differentiation. But here he took a unique approach, and he started to look for long non-coding RNA changes in gene expression that might be involved in not just neuronal lineage specification, but also might be disrupted in neurological or neuropsychiatric diseases. And he discovered one such link RNA that seems to be associated with a neurodevelopmental syndrome. And it really was like a paradigm when I read his paper about how you can integrate human genetics and then sort of functional testing, looking at neuronal lineage in induction. So that was an exciting paper. Then he's really been at the cutting edge of spatial transcriptomics at Harvard for his postdoc. Looking forward to seeing the latest. Cheen Euong, go for it.
Hey everyone, thanks for coming, and thanks for the nice introduction. So my name is Cheen. I'm a postdoc in Xiaowei Zhuang's lab, and today I'm going to talk about papers that recently published on developing a methods to interrogate the epigenome. So we call it epigenomic mFISH. So as you all know, during development, mammals develop from a totipotent zygote into pluripotent ES cells, multipotent progenitors, and subsequently into different mature cell type. And all these cells share a same sets of genetic information in the form of DNA. So the genomic DNA are read in the context of cellular histories and also signaling pathways to form like distinct chromatin states. So it can be permissive or it can be repressive chromatin states. So these chromatin states dictate what gene can be turned on, where the gene can be turned on, and which gene to be turned on. And the chromatin states can be marked by distinct different histone modification. So one of the well-known one, like this is published in 2001, where they describe like this very, very well-known histone marks K4 trimethylations that mark active promoter. And another example of the very well-known histone marks is K9 trimethylations that marks constitutive heterochromatin. So because of that, there are multiple different sequencing techniques has been developed to actually interrogate like the sequence, where these histone marks are located in the genome. But obviously, sequencing technique always involves disruption of the cells. So basically, the cellular information and where the epigenomic marks are located within a certain tissue are gone when you dissociate the cells. Multiple groups have started to develop imaging techniques to actually to preserve the cellular context of those genomic loci that have epigenomic modification. So one of the techniques that was developed and reported recently is called Scepter. So Scepter uses three different components. So we have like an antibody recognizing one histone marks and another ones that recognize another histone marks. And when they probe the genomic loci of interest with fluorescently labeled oligonucleotide, they can actually localize that and image them, and then analyze the relative distance of this oligo probe, labeling the genomic loci to any of the two histone marks. And doing comparison, they can see whether this genomic loci is either active or inactive. And in the paper, they describe this technique in three different loci, in the two different histone marks. So you can get kind of like the conclusion that the techniques can only interrogate like 3 loci, which is probably not enough. And the other papers that was published almost 10 years ago now, that they use similar techniques, proximity pretty much a proximity ligation followed by amplification. So you use primary antibody, secondary antibody, but the difference is the secondary antibody have oligo. Both have oligo nucleotides. So when they're in close proximity, they can form a template for rolling cycle amplification. And then using that, they can actually localize the signal by virtue of proximity. But this technique obviously suffer from low dynamic range, pretty high background, as you can see over here, and then also very low throughput. So in that paper, they only describe the technique to be performed on one single epigenetic loci. And more recently, there's a more high throughput methods described by Long Kai and co-workers, where they use similar ideas. So basically use oligos to tag different genomic loci and the bin size of 1 megabase resolution, and then coincide the individual genomic loci with different histone marks. So in this case, shown over here is Th3K9 acetylation. So they did about 3,600 loci on a 1 megabase genomic resolution, and some of them at a lower resolution and overlap with the antibody. But one of the downside of these techniques, this kind of overlap by signal by fluorescence, immunofluorescence, is that most of if you perform sequencing before and analyze all these histone marks, the peaks are relatively tiny. So they're about 100 base pair long. So by just getting a very information from 1 megabase, you actually kind of integrate the signal across 1 megabase pair and all the information are lost. So the idea when I first started grad school is to develop a technique that can solve the problems of the number of loci that can be interrogated and also the genomic resolution. So we come up with a technique called epigenomic mFISH. So when I joined Xiaowei's Zhuang's lab, Steve Hanikoff just published a technique. So it's a technique called cut and tag that uses a very interesting fusion protein called PATN5. So it has a protein A that binds to antibodies and a TN5 that can be loaded with different loaders. So when the protein A recognizes antibody, it can tag the genomic DNA in close proximity. So we use the PATN5 developed by Steve Hanikoff in our protocol. So this is the workflow of epigenomic mFISH. So first off, we have epigenomic marks of interest. So it could be K4 trimethylation, which marks active promoter, or K27 acetylation, which marks enhancer. So by using primary antibody that recognizes the marks and secondary to amplify it, we can subsequently treat the cells with PATN5 loaded with specific loaders. So then, because of the proximity, the TN5 will tag into the genomic DNA they're in close proximity with the histone marks with the tag carried by TN5. So the tag there are incorporated. In our case, we actually added a T7 promoter sequence. So because of that, subsequently, the genomic DNA can be amplified by T7 polymerase into mRNA signal. And so basically that's a steps of amplification. So this amplified mRNA can be reverse transcribed and sequenced, or it actually can be detected by mFISH imaging technique. So what is actually mFISH? So mFISH stands for multiplex area robust fluorescent in situ hybridization. So it's pretty much a FISH technique with an area robust or area correction technology incorporated into it. So that kind of enables parallel interrogation of 10,000 species of mRNA within the single cells. So in this example I'm showing over here, in orange is the transcribed mRNA, and then we introduce the probe. So the probe have a complementary region to the mRNA of interest. So this set of probe recognizes the mRNA produced by this loci. And then by imaging it in sequential imaging rounds, for example, in the second round, we get like a positive signal because there are the oligo carrying fluorescence oligos binding to this region, we get a signal. And then in bit 5, in round 5, we get a signal, and so on and so forth. All these distinct rounds of fluorescent signal can be decoded into a particular loci. So basically that enable this parallel interrogation of hundreds of loci, with each loci carrying different on and off information. So because of that, we can now have a protocol that you can actually kind of benchmark. So the first things that we decided to test is whether the signals are reproducible. So we do a reproducibility test, and it showed that actually the average number of decoded spot per loci per cells are correlated between the two samples. That is good. And then we next ask the question of whether the signals are depending on the antibody that we use. So basically we compare the K27 acetylation in this case to the IgG. And sure enough, when you compare the average number of decoded spot per loci per cells, on the condition with the antibody, we have more decoded spots per cells compared to the IgG. And then one of the things that people are interested in, at least in the epigenomic sequencing world, is whether you can detect like this, how efficiently you can detect it. And so we decided to perform these types of tests using K4 trimethylation against candidate loci of the promoter regions of housekeeping gene. So we use 50 candidate loci from housekeeping gene. And when we do the assays, we show that actually 20% of the cells have one decoded spot per loci, and some of 10% of the cells have more than one decoded spot per loci, meaning that when you add these two together, it's about 30% detection efficiency, which is not too far away from the sequencing counterpart, which can range from 8% to 40%. Now that we have these two, now that we know that we can actually do epigenomic mFISH, the other questions that people are asking, will ask is whether when you perform the assays, this subnuclear localization information is preserved. So to answer the question, we perform the epigenomic mFISH individually against the 3 marks and co-stain it with a nucleospeckle marker and measure the distance of individual spots to the nucleospeckles. And sure enough, when we measure that, the 2 active marks, K27 acetylation, which mark the active enhancer, and K4 trimethylation, which mark active promoter, are closer to the nucleospeckle compared to K27 trimethylation, which is an active mark. This is in accordance with the role of nucleospeckle in coordinating active transcription. So now that I have a workable technique in cultured cells, we next wonder whether we can actually perform it in, obviously, brain tissue. So one of the things that we thought about performing is in embryonic brain using K4 trimethylation. And we curated 127 promoter loci. So those are the promoter for transcription factors with well-defined expression pattern in embryonic brain and adult brain. So when we perform that, we get this beautiful composite picture of all the decoded spots. As you can see, there are gradient of different expressions showing that different transcription factors have different expression pattern. And when we zoom in, you can see that the individual spots are falling within the nucleus, indicating that we are actually detecting the correct signal. So this is a range of examples that I'm showing here by comparing the gene, so basically marked by in situ hybridization obtained from Allen Brain Atlas, and the corresponding K4 trimethylation signal of the loci, the K-FETZF. So as you can see, the epigenomic signal over here actually coincide with the mRNA signal as indicated by in situ hybridization. And if I were to take another example, FOXG1 over here, FOXG1 have a very, very characteristic expression in the cortex and also in the striatum. As you can see over here, both the epigenomic mFISH and the ISH show very, very similar pattern, indicating that indeed, when we measure the activity of the active promoter of a particular gene, the pattern actually matches the gene expression pattern, showing that actually the epigenomic mFISH technique that we developed also work on tissue. Now we decided to challenge ourselves on a totally different question. So we asked the question of whether we can actually go all the way to single cell in complex tissue. So whereas in embryo, all the nucleus kind of overlaps together, making actually segregation of the nucleus very, very difficult. In adult, the nucleus are very, very well separated. So, and they are organized in distinct layers. So we can also test whether the pattern that we detect actually matches the layer-specific patterns that we know in the literature. So shown here is a composite pictures of all the dots that are decoded in adult cortex. And as expected, when you zoom it in, all the spots fall within the nucleus. And then when we do a systematic comparison across all the loci that we are interested in, so it's about hundreds of loci, and also corresponding RNA mFISH signal, we have a very, very high correlation efficiency. And then when we take a closer look at individual example, we can see also layer-specific enrichment. For example, RNA mFISH for COOX2 form a very, very characteristic layer 2, 3, and 4 expression pattern. So basically that shows the mRNA expression. So when we see the corresponding signal for epigenomic mFISH for COOX2, they also specifically labeled layer 2, 3, and 4. So kind of matching the what you would expect. So now that we established that we can actually do it both in complex embryonic tissue and adult tissue up to a single cell level, we next want to understand whether this is specific to this antibody or whether the technique can be applied to different histone marks as well. So we decided to do it on K27 acetylation. So K27 acetylation is a mark for labeling active enhancer. So when we work on the projects, there's not too much information for layer-specific K27 ChIP-seq data available for layer-specific enhancer, but we did find a paper from Allen Brain Necklace where they performed ATAC-seq using fake solid cells. So what they did was they have a transgenic mouse that bred, so basically a specific transcription factors promoter driving CREERT2, and they bred it with an AI14 mouse. And using that mouse, they are able to actually mark cells in different layers. So when we do a microdissection and then dissociate the cells, fake sort the cells, and then subsequently do a TN5 reaction, so ATAC-seq sequencing on those cells, they can get a layer-specific ATAC-seq profile for individual layers. So because ATAC-seq can overlap both with the active promoter and enhancer, so we peaks that are actually away from the promoter, and we peak like 139 loci from those layer-specific ATAC-seq peaks. And we perform epigenomic mFISH on adult brain using the library that can detect this number of peaks. So when we do that, we get a specific profile, and shown here are 3 different examples. So this loci 1, 23, 24, and 27 are 3 different loci with some epigenomic signals in layer 2, 3, and 4, although you can see that the relative relative ratio is the relative difference between the 3 different loci. So when we take a look at the corresponding ATAC-seq signal, it shows similar enrichment in layer 2, 3, and 4 for all these 3 loci. So next, I wonder what genes are actually in vicinity with these 3 different K27 acetylation peaks. So indeed, we found Euong5D, and they all fall within a subtet in this within this major tet over here. So when we look at the corresponding expression patterns for Euong5D, you can see that the Euong5D RNA-ish and also the RNA mFISH both show layer 2, 3, and 4 expression, suggesting that these 3 epigenomic, these 3 peaks actually are potentially regulating the expression of Euong5D. And we also have a summary results for all the peaks available. And when we do a systematic comparison between our results and the ataxic results, we see a very strong correlations between the two, implying that the technique indeed can actually recapitulate what has been described before in the literature using sequencing methods. And finally, we decided to use this technique to answer a few questions that we are interested in, especially with regards to enhancer biology. So I mentioned that K27 acetylation marks the enhancer, whereas for promoter, most of the time you can actually predict how the promoter activity would look like in cells, because it would probably resemble the corresponding gene. But for enhancer, it's much more difficult. So we decided to use this to answer the question of enhancer promoter interaction or enhancer gene activation in types of interaction question. So to do that, we obviously curated again 150 loci using the sequencing data from ENCODE, where they have like a distinct enrichment in 4 midbrain or high-brain enrichment. So we used those 147 loci, and then we decided to perform epigenomic mFISH against them. So for example, the first thing that we checked was whether the signals that we obtain can actually recapitulate the gene expression pattern. So because we're measuring the enhancer activity, we want to see whether the enhancer activity is similar to the corresponding gene expression. So these are the predicted pair by Bing-Ren's group. So in the paper, they predicted that, for example, this loci over here actually is actually regulating the Neuro G2 expression. Sure enough, as you can see over here, when we see the epigenomic mFISH signal for this loci, it closely matches the nearby gene Neuro G2, which is previously predicted. And the same goes to EMX2. It has that expression in the cortex. And again, like you can see the corresponding expression in the cortex. And next, we asked the question of whether the technique can be used to also infer a new gene enhancer gene pairs. So we decided to look into the peaks and expression, the activity, and see whether there are genes that actually matches the expression pattern. So as you can see over here, loci 95 form this distinct pattern in the cortical plate of the cortex, and also a distinct patches of activity between the striatum and the diencephalon. And you can see that matches the nearby gene promoter TBR1. And the same can be seen for FOXG1, where loci 31 show this distinct pattern. And then the closest promoter to loci 31 is FOXG1, and FOXG1 also show the same pattern. And this is particularly encouraging. But so we decided to do more genome-wide, more of a generalized characterization, where we cluster the field of view. So basically, is this all aggregate signal of all the field of view that we have of the data, and all the different target loci, and then we do a hierarchical clustering. So by doing that, we can actually cluster all the target loci that have a similar pattern together. And we found like 6 distinct clusters. And when we take a closer look at all the distinct cluster, we can see that actually this blue cluster over here with this high expression pattern, this particular field of view actually is in the midbrain. And when we do a systematic characterization, you can see that actually distinct cluster actually have like different enrichment in different part of the brain. And one of the clusters is particularly interesting, like we call it cluster 4, that labels the proximate region of the developing brain. So we discovered that actually there are sets of 10 different enhancers, and in the close vicinity of this gene over here, so TCF7L2. So TCF7L2 is a gene that is downstream of Wnt signaling. So as you can see over here, there are enhancers that are closer to another gene called VTI1A. But when we take a look at the epigenomic mFISH pattern, all 9 of them, the enhancer that we're interested in that falls within this genomic loci over here, they actually all have similar expression pattern or activity to the gene expression. So basically we can actually kind of tie the enhancer, all this 9 enhancer to that, and kind of tie it to this gene and associate like this enhancer actually regulate the expression of this gene. And obviously, this is kind of intriguing, right? Because this enhancer actually is in very close proximity with the promoter of another gene. So traditional like computational algorithm would have actually, by definition of the high C, would have tied the enhancer to regulate the close by gene, which is VTI1A, but that is not the case. And also, curiously, when we do a motif analysis on the sequence, DNA sequence of all these 7 region over here marked in green, we actually find back the TCF7L2 motif, seemingly imply that this coordinating sets of 9 enhancer actually is what they call in the literature redundant enhancer. So basically they are redundant in a way that, because to ensure the robustness of the expression of T7L2. So with that, I would like to end my talk. I would like to thank my boss, Xiaowei Zhuang, for the support and the advice during the whole publication process, because it's during COVID time, and my close collaborator, Tian, and all the Zhuang lab for inputs. I would like to also thank my funding sources, Sufri, for the support, and also the Pathway to Independent grants that they gave me recently. And thank you for your attention. And I would love to listen to your feedback.
Thanks so much, Cheen. Great talk. We're open for questions. Just maybe I'll start with one question. I was sort of wondering if you have experience with this yet, or your thoughts on how stable some of these peaks, some of these epigenomic changes will be across different aging or different states of the animal, compared to compared to the transcriptomics.
Yeah, so obviously the epigenomic signal seems to be dynamically regulated. So there's been, at least in the ChIP-seq, when they perform like ChIP-seq sequencing across different ages by Bingren and Exowiso and co-workers, they show that actually the signal can actually be dynamically regulated, so things can actually goes up, and so there are some things that actually, some enhancers that are very important in regulating some cellular processes in embryonic, doesn't necessarily regulate the same thing in adult. So there are definitely dynamically regulated. We did actually use the same library to measure the patterns for like, for example, the 10 loci that are shown at the end in the adult. And interestingly, they have the same pattern, so they still show a very midbrain enrichment, but they are split into 2 different parts. So basically, I feel like this technique is kind of interesting because it can actually identify enhancers that actually kind of evolve into, so it's kind of like a lineage tracing technique. So the enhancer can actually mark the cell differentiation over time also, if the epigenomic signal actually preserved during differentiation.
Got it.
Yeah.
And what's the prospect of doing this on human?
Yeah, so so I think I currently don't have plan to do it on human because the human tissues are hard to come by, but I do envision it to be similar in difficulty to do it. There are people in the lab that have performed mFISH on human brain, and so basically not epigenomic mFISH, but RNA mFISH, and they work well and comparatively well. So I think if I were to apply this technique, it would have little difficulty, but we haven't done it because I think human brain is really requires some sort of hypothesis to be, and it's also very difficult to obtain from the lab. Yeah.
Got it. There's a question from Xing Wei asking, how many enhancer loci can you detect in one round?
So right now the technique, so when you say run round, is one experiment pretty much. And for one experiment, we detected up to 200 loci right now in the context of tissue. In cell culture, we have attempted to detect up to 2,000 loci, enhancer loci, but we haven't published a paper. So I think that is something that we are currently actively working on. So it actually just to increase the throughput of the technique. Yeah.
Got it. Okay, Cheen, awesome talk. Love to collaborate with you on this. Really exciting. And
yeah, thank you so much.
And Zhejiang, do you want to introduce Peng?
Sure. Great. It's my great pleasure to introduce Professor Peng Shi. She's a professor in the Institute of Translational Medicine in Zhejiang University, just in Hangzhou, not far from Shanghai. So Peng has interesting career path. She was trained as a physician, so in Thai Medical College, and after a couple of years of medical practice, she decided to start her career in scientific research, beginning by a PhD in UT Texas, Ontario. After PhD, she had a postdoc at University of Florida, working on immunology. She started her own lab in the beginning in the US and Cedars-Sinai College in LA. After a few years, she come back to China, set up her own lab in Zhejiang University, Institute of Translational Medicine. She focused on interesting cross field in the immunology and neuroscience. So has interesting discovery about the role of microglia in a series of immunologic disorder as well as the neurological disorder. So today, Peng will share with her recent study about the role of microglia in sympathetic nervous system. Okay, welcome, Peng.
Thank you so much.
Sure.
Let me share my slide first. Yeah, everyone can see my slide.
Sure, take it away. Yeah.
Cool. Thank you. Thank you so much, Zilong and Aaron, for this opportunity to let me to share our work with the Neuroscience Society. I'm going to talk about how our understanding how microglia, the resident microglia in this brain, how to participate in neuronal activity, which further modulate the sympathetic outflow and cardiovascular function. As mentioned, I'm in Hangzhou. We just passed the most beautiful season, but actually any time in Hangzhou is very beautiful. Welcome to Hangzhou. So let me start. So this picture was taken in Yalta Conference in 1945, 70 years ago. That's right after the Second World War. People in this snapshot were the most influential leaders by then. Beyond their powerful people in the world, they also actually, all of them are hypertensive patients. 2 months after this conference, actually President Roosevelt collapsed and died of a cerebral hemorrhage. Today, healthcare and professionals and even public, everyone knows and understand the importance of proper diagnosis and treatment of hypertension. By then, there was no effective medications to treat elevated blood pressure. So cardiovascular disease is the leading cause of death, overriding cancer. However, compared to the high burden of cardiovascular modality and mortality as the leading cause of disability and death, there's a relatively limited pipeline of new drugs for cardiovascular disease. If we break down all the risk factors, and you can tell that hypertension actually is the leading major contributor, which result in over 400,000 deaths per year in the United States. Actually, in China, the hypertension is also a major prevalent disease. Over 30% of adulthood are diagnosed as hypertension. Most striking, data from this survey, you can tell the people who has managed their blood pressure within the normal range, which is below 140 over 90 millmercury, is about 5%. So it's a very low numbers. Hypertension is also called the silent killer treatment of hypertension can dramatically decrease stroke and the related health disease. As we know, the dementia is the major health issues with the aging. So lower the blood pressure actually can decrease the dementia progress. So in the past 70 years, two breakthroughs have already contributed significantly to reduce the blood pressure. First is identify risk factors for hypertension, such as we know we need to decrease the salt intake. Second is the development of a safety and effective anti-hypertensive drugs, such as the ARBs or ACE inhibitors or calcium channel inhibitors or diuretics. In clinical setting, we know there's roughly 10% of patients are estimated to have resistant hypertension, meaning these patients are refractory to current medications. So the physicians took a very aggressive approach, which is by denervation of the renal sympathetic nerves and then causing the renal vasodilation, and thereby can lower the blood pressure. And this invasive treatment, as you can see here, that's from the patient data. And after the renal nerve denervation, they have a dramatic decrease of blood pressure compared to the sham control patients. So this suggests that the sympathetic tonicity is a major contributor to hypertension development. So according to Ohm's law, the blood pressure is determined by the cardiac output and the peripheral vascular resistance. Cardiac output is defined by the cardiac stroke volume and the heart rate. All this actually controlled by the sympathetic nerve activity. So sympathetic nerve activity is generated by a couple of nucleus within the brain. And in this sagittal view of a mouse or rat brain, I highlighted a couple of nuclei, particularly the PVN in the hypothalamus and the rostroventrolateral medulla, that's RVLM in the brainstem. And those are directly contribute to the sympathetic outflow. And my study actually is focusing on the hypothalamic PVN. It's not only because it is upstream of RVLM, and also we know in addition to the presympathetic neurons, they also contain the vasopressinergic neuron and the CRH, the cortisol releasing hormones. So PVN is a critical nuclear to maintain the body homeostasis and also respond to the environmental stress. So the question is, what cause hypertension is characterized by the increased sympathetic nerve activity and chronic low-grade steroid inflammation, non-steroid inflammation. Back to at least a decade ago, a couple of groups started to investigate the correlation between the sympathetic nerve activity and inflammation. My postdoc work showed that ICV infusion of minocycline, which is a second generation of tetracycline, can fully abolish angiotensin II induced hypertension, and as well as lower the plasma norepinephrine. As I mentioned, plasma norepinephrine is a hallmarker for the sympathetic excitation. So the question is, what causing this inflammation in hypertension? So in the past decades, more and more studies emerged and showing that microglia actually is the primary immune cells in the brain play a critical role involved in cardiovascular disease, which may bridge the central nervous system with the peripheral immune organs or tissues together, which aggregates the tissue organ damage and therefore impaired cardiovascular function. So let me briefly review microglia. As the audience may know very well, the microglia has been shown to support multiple sympathetic same as homeostatic functions. For instance, microglia guide the neuronal circuitry formation in development, and contribute to the synapse formation, pruning, support the neurogenesis, myelination in the adulthood. And recently, two papers studies demonstrate that microglia also associated with the cerebral capillaries to modulate the cerebral blood flow. So, what happened to microglia along hypertension development? As you see here, all the microglia we 3D reconstructed in different brains. That's 3 days after hypertension induction, the microglia undergo the dramatic morphological changes, particularly in the microglia in the PVN. And we want to ask whether microglia is important has a cause effect with hypertension development. Therefore, we use the CD11B promoter-derived human diphtheria toxin transgenic mice. By ICV infusion of DT, we can specifically deplete microglia over 90% with minimal impact on neurons or astrocyte, at least in their distribution aspect. And then in the first experiment we did is in 2 cohorts of CD11B DTR mice, we induced hypertension by angiotensin II. After blood pressure steadily go up, in the 1 cohort of mice, we deplete microglia by ICV infusion DT. As you can see here, without microglia, the blood pressure didn't go as high as the control cohorts. The second experiment we did is in 2 cohort of mice, we deplete microglia in baseline in the resting state, and loss of microglia didn't change the baseline BP. However, once we induced hypertension by angio-2, you can see the mice without microglia actually develop hypertension more promptly and more dramatically. So in the past few years, many elegant work has indicated that microglia in surveillance state negatively modulate neuronal activity. This study is focusing on the neurons in the striatum or cortex. So whether microglia regulate PVN neuronal activity by the same mechanism or not. So that's what we are trying to figure out. So we asked, do microglia regulate sympathetic outflow via the modulation of the presympathetic neurons in the PVN? And then we back to use the CD11B DTR mice again. And without depletion of microglia didn't change the baseline resting blood pressure. However, the mice displayed the tachycardia and attenuated the heart rate variability, as well as the baroreflex sensitivity. And meanwhile, we found the microglia depletion increased the plasma norepinephrine and epinephrine. As I mentioned earlier, those are hallmarkers for the sympathetic excitation. To analyze the PVN presympathetic neuronal activity, we did the PET clamp by pre-labeled RV-LAM projecting PVN neurons with fluorescent retrobeads. Removal of microglia didn't alter the resting membrane potential and resistance, but there are more spontaneous firing neurons in the labeled PVN neurons. Meanwhile, we found the firing frequency is dramatically increased. It also prolonged the duration of each spike. So given the voltage-gated potassium current are very critical in controlling the neuronal firing pattern, therefore we analyzed the depolarizing outward key current. Surprisingly, loss of microglia significantly suppressed the outward potassium current, particularly the transient A-type current, we call the IA current. And then to understand the molecular mechanism, we did the single-cell RNA-seq by comparing the transcriptome of PVN presympathetic neurons with or without a microglia, surrounding microglia. We found actually there's less than 400 genes changed. And among the differentially expressed genes, we focused on the ion channel related categories and found that the genes coding the potassium channels are changed dramatically compared to the other ion channel categories. Given that the IA current is suppressed the most, we checked the 6 genes coding the alpha subunit of the key current, and only KCND3. This gene is significantly suppressed after microglia depletion. And this result is validated by the in situ hybridization. And then to understand what is how microglia modulate the neuronal KCND3 expression, we first tested in vitro and in culture cells by culture the neuronal cell line PC12 with or without the BV2, which is the microglia cell line. And you can see the significant increase the KV44.3, which is the protein product of KCND3. And this increase also occurs in the transveal co-culture, suggests that microglia regulate the PBN, the neuronal KCND3 expression by a paracrine mode. And then by checking we visit our bulk RNA sequencing data. We narrow down to the 28 genes coding the secretable proteins in the top 5% of highly expressed genes in the homeostatic hypothalamic microglias. PDGFB actually is among them. Meanwhile, we check another 2 studies using a single cell RNA sequencing, and they've also show that PDGFB is among the top highly expression genes in their data set. And more importantly, we found from another bulk RNA sequencing data, we found that in adulthood, microglia is the primary cell type expressed PDGFB. So, and in contrast, their counterpart of a brain macrophage actually have barely expressed the PDGFB, as you can see from this FISH immunohistochemical co-staining. And next, we crossed the CXCR1 Cree ER mice with the PDGFB flux mice, and offspring were treated with tamoxifen to induce the depletion of PDGFB from the microglia, and which resulted the neuronal KCND3 expression reduction. And meanwhile, we found that the reduced key current, particularly the IE current, as well as the dysfunction of the autonomic dysfunction, which is mimic what we saw in the microglia depletion mice. And we also tested the receptors in the neurons, which subtype of the neural receptors mediate the PDGFB effects. As you can see here, depletion knock down the receptor alpha instead of receptor beta actually show the similar effect as the ligand depletion from the microglia, suggest that microglia release the PDGFB through binding with the receptor alpha and promote the KCND3 expression in neurons, and therefore maintain the sympathetic tonicity, therefore influence the blood pressure. And next, we want to ask, does this pathway is also applied to hypertension? And when we check the literature, actually we found there's a cancer patient, if they receive the tyrosine kinase inhibitor treatment, such as sunitinib, and they have a better chance to develop hypertension. And this blood pressure climbing is irrelevant with their hypertension history or not. So once they're on this drug, this tyrosine kinase inhibitor, this TKI, they have an increased blood pressure. And once stop taking this drug, and the pressure goes down. And another review showing that in this family, the tyrosine kinase inhibitors, there's a couple of drugs, they all have been reported have in different numbers of the hypertension incidence. And we're focusing on the pazopanib, and we want to know whether this pazopanib, the TKI, can induce hypertension in animals, because just now we talk about the PDGF receptor. Actually, they belong to the tyrosine kinase family. So as you can see here in adult C57 mice, IP injection of pazopanib elicits sympathetic excitation and hypertension within a week. And this is associated with a reduction with the KCND3 expression in the PVN presympathetic neurons, and also they suppress the outward K current. And in summary, actually, we just shown that the microglia they producing and releasing the PDGFB, and by binding with the receptor in the neurons, the receptor alpha on the neurons, and promote the KCND3 expression and maintain the normal sympathetic outflow and blood pressure. And the disruption of this pathway may as a mechanism to leading to the essential hypertension. At the end, I'd like to thank all my lab members, and particularly Cheen, who is the leading author for this, the work I just presented. And also I want to thank all my colleagues in Zhejiang University and the affiliate hospital, affiliate hospital, and the Zhejiang Hospital, and also my research support grant. And at the end, I like everyone. Hopefully, once the pandemic finished, we can join offline conference. And welcome to Hangzhou. Thank you.
Okay, thanks, Peng. Looks like Zhejiang's frozen. Awesome talk. We all look forward to visiting you in Hangzhou. Chuan, do you want to ask a question?
Hey, Aaron. Hey, Zong.
I'm sorry, yeah, come back again.
Great talk, Peng. Just to have a couple of questions. First of all, is how you actually specify the channel with Kv4.3, because obviously the depolarization on the neurons could cause by the other channels as well. How can you be, because I don't see any data which you condition knockout, Kv4.3 on the neurons to abolish the effect. How can you be so sure this is the dominant effect through this channel?
Okay. Let me share my slide again. Actually, we did I didn't give enough background. Actually, IA current has been reported being a very critical current to maintain the PVN sympathetic neuronal activity, at least in the PVN neurons. And we did. There's one. Here, you see here. Actually, we reinforce the KCND3 expression by retrograde inject the creep in the RVLM, and in the PVN we injected the fluxed KCND3, human KCND3 in mice without microglia, which can fully restore the key current. So suggest that the microglia depletion cause the effect through the KCND3.
I see.
Yeah.
Did you do any, molecular mechanism work on the PDGFB on the neural expression Kv4.3?
Yeah, actually, that's what we're doing now. We mining our RNA sequencing data from the neurons, actually we trying to first from the receptor side, the downstream target. Let me see. Let me share it. The receptor downstream signal is very complicated. Actually, it's here, and I made the long story short. The ligand actually can be a different combination. Just now I propose it is B. Actually, they can be dimerized as BB or AB, and the receptor is alpha or alpha beta, or they can be heterodimer alpha beta, and the signal is not here actually. They can be MAPK and MAPK-ERK signal. And we found it's very difficult to start from the receptor, and we're trying to figure out what really changes, like the transcription factors by deplete microglia PDGFB or knock down the receptor alpha. What happened in the neurons? And then we check the KCND3 in the promoter region of the KCND3, whether the altered the changes transcription factor is important for the KCND3 expression, and therefore we can try. We want to link to the receptors and kind of a backtracking what is the signal, intracellular signaling from the receptor down to the KCND3 expression. Here I didn't show. Is here, and there's two papers actually back to 1990s. In OPC, actually they have been reported. These only two papers we found in the literature. The PTGFB actually can promote promote the potassium channel, but that is not the IA current. So I think they have cellular specificity for in different neurons. Yeah.
And this receptor is only enriched in the neurons?
Actually not. Actually not. This receptor is highly expressed. If you see here, the red, is very bright red. This is the OPC. PTGFB receptor alpha actually is highly expressed in in OPC, but we checked from the sequencing data, and it is way less in the neuron compared to the OPC, but they have expression around 8.9.
Sorry, go ahead.
Yeah, sorry. So that's why we did the staining and 3D reconstruct the PTGF receptor in the neurons, in the labeled neurons, PVN neurons, and you can see here there are expressions inside the neurons, but definitely OPC is highly expressed the receptor alpha.
The reason I'm asking is because the PDGF receptor signal, it's have kinetic and non-kinetic signal pathway, not necessarily through the transcription regulation, because in the peripheral, if you look at the sympathetic neurons, these actually receptor bind with the ligand can internalize and then to trigger and then translocate it as into the lysosome. It's actually have different signaling pathway, not necessarily directly connected to the direct transcription regulation. Maybe you can, take a look into that just in case. But yeah, awesome talk. Thank you.
Thank you so much. Thanks for your suggestion.
So I have a quick question about the very interesting microglia activating neurons, and so what happened then? So after PV neuron being activated, so we will be releasing something like AVP to regulate the blood pressure afterwards.
Actually, no. There are. Let me find my.
Yeah, how they end up regulating the blood pressure after that?
Okay. I didn't. I should give the sympathetic nerve activity actually is through the. We call this the motor. The neurons in the RV lamp we call the vasomotor neuron. can you see the slide? Yeah.
Sure. Yeah.
So the neurons here in the RV-LAMP, we call the vasomotor neuron, means their depletion of these neurons, causing the blood pressure dramatically decrease about 40 millimmercury. So it means the neurons in this pathway actually contribute to the tonicity. So these neurons will go to the vessels, the heart. Yes.
Yeah, sure. Oh, there's one more question on the chat bar. You can see that since I'm on my cell, so how to get to the
these PVN RV-LAMP neurons release a neuropeptide. Actually, so far we know they can be CRH positive neurons, but not oxytocin neurons. And there are some studies working on showing that there's different categories of PVN neurons also releasing the oxytocin, but that's most of chance we're not label it, because in our single cell RNA sequencing data, we found no AVP or oxytocin expression in the labeled cell, but there are CRH expression.
CRH, okay, amazing. Okay, that's very interesting.
Yeah.
Great.
Can I ask a question?
Yeah, sure. Go ahead.
Yeah. So very, very striking results. Very amazing. When you deplete astrocyte, you see a very dramatic effect on the blood pressure and also the activity. I was wondering, I maybe missed, how long did you wait after you deplete the astrocyte to measure those changes? I was wondering whether other cell types might also have similar. So astrocyte clearly play a role. You're showing very, very strongly. But what about if you waited longer? Are the other cells can step into also to compensate for the loss? Because like PDGF B is also expressed in other cells like blood vessels, smooth muscle cells, fibroblasts. So there are other sources of PDGF B.
To the microglia, actually, we from the RNA sequencing data, we found there's in adulthood they are highly expressed in neuron in microglia. Let me share. See, can you see my mouse? Here. This study actually using it's a bulk RNA sequencing data and compare the different cell types in the cortex, I think. And actually you can see microglia is dominant cell type to produce the PDGF-B. And the second resource may be the endothelial cells. Actually, that's very interesting. PDGF-B as a paracrine factor or a molecule, and mostly people believe that is endothelial cell derived. But because first we think maybe in adulthood, because the tight BBB, so the endothelial cell produced the PDGF-B may not can diffuse into the parenchyma of the brain, and therefore I think all the PDGF-B from the microglia maybe is the major source. But after microglia depletion, whether other cell type can compensate this loss, it could be. We didn't test it because we only used the mFISH, and this depletion can be up to 5 months. That's what we test. So up to 5 months.
I see those changes are observed 5 months after the depletion.
Right.
That's cool. Yeah, that's great, because hypertension is it's a chronic disease. So I'm interested in whether the effect is long lasting. That's amazing.
Yeah.
Congrats.
Thank you.
Great. All right. Great. Thanks, Peng and Cheen, for amazing talks today. Thanks, everybody. So, Aaron, so enjoy Philadelphia.
Yeah. Thanks.
Yeah, come back,
everyone.
Yeah.
Thanks, Peng. Thanks, Cheen.
Thank you.
See you next week.
See you guys later.
Bye.